Although the theory of panspermia gives us an understanding of how life developed on came to Earth, it does not answer how did life originate, whether it is outer space or whether it is on Earth. So even if we believe that life has come from outer space onto the Earth, but still, how did that life develop in the some part of the outer space? So the origin is not known, but strictly speaking, the theory of panspermia is not a theory of origin. It is only a theory of transfer of life from one part of the universe to the Earth. Now, even if we believe in this theory of panspermia, we still have to answer the question, how did life develop in that particular part of the universe before it came to the Earth? So the question still remains unanswered. So this last and fourth theory which I am discussing here is the theory of chemical evolution of life. So this theory tries to answer whether it is Earth or any other part of the universe, how did life originated there. So this theory was proposed by these two people here. One is from England, so he is Oparin, and this bald man here is from no, Oparin is from Russia. This bald man here is Haldane. So Haldane is from England. So one Russian and one British scientist. So these two fellows put together a theory called theory of chemical origin of life. Now, what does the theory say? The theory says that life originated through chemical reaction. So they propose that the various elements which are there in the Earth's atmosphere, so they combined to produce simple molecules. So these simple molecules, they gave rise to complex molecules, and these complex molecules, they got aggregated or clustered to give rise to primitive cell. So once the primitive cell developed, then this primitive cell evolved by means of organic or biological evolution to give rise to all these present day multicellular, different types of multicellular plants and animals. So this theory is nothing but it tells us how the non-living elements, simple molecules gave rise to a living structure called cell, and all this was by means of chemical reactions. Now these chemical reactions, which led to the formation of the cell, it took place when Earth was very young, and it was also very violent when it was young. So this is a picture of how the Earth looked when it was still in the process of developing. So this is the picture of violent Earth. So what are the activities that were taking place on this very primitive violent Earth? So on this primitive violent Earth, there was lightning, there was volcanic activity, there was incessant rain, okay, rain which went on for hundreds and thousands of years continuously. Now for rain you need water. So initially Earth's atmosphere had oxygen, but it combined with hydrogen to produce water. So that is why initial atmosphere of the Earth did not have any oxygen, because all the oxygen combined with the hydrogen to form water, and that led to formation of reducing atmosphere. So reducing atmosphere is one where there is no oxygen. So this makes it very necessary for the first living organism or cell to evolve on earth to be an anaerobe, because since initially there was no oxygen, the organism cannot depend on oxygen for life support. So it has to be an anaerobic cell or organism. And also this primitive atmosphere had gases like methane, ammonia, hydrogen, and water vapor. So all this is what Oparin and Haldane said in their theory of chemical evolution. So they said that life evolved due to chemical reactions, and the conditions on earth were like these when the earth was very young. So under these conditions, they said that the first primitive cell originated from which the rest of the organisms, plants and animals evolved on earth. Now this had to be demonstrated. So a scientist called S.L. Miller. So S.L. Miller is an American biologist. So S.L. Miller demonstrated the chemical evolution theory proposed by Oparin and Haldane. So what he did was basically he would replicate the conditions of primitive earth. So S.L. Miller replicated the conditions on primitive earth as proposed by Oparin and Haldane in their, so their theory is known as chemical evolution theory. So in order to replicate the conditions of the primitive earth, S.L. Miller uses a special apparatus. So this is known as Miller's apparatus. So a schematic diagram of this Miller's apparatus is as shown here. So this is a closed flask. So the apparatus taken by S.L. Miller is actually a closed flask apparatus with two bulbs. So one large upper bulb and one small lower bulb. So in the large upper bulb, Miller took the primitive gases like methane, ammonia, water vapor, and hydrogen. So these primitive gases were taken in the upper large flask. So this upper large flask, it represented the primitive atmosphere of the primitive earth. Remember that there is no oxygen taken because the primitive atmosphere was reducing atmosphere. Now in the lower small bulb, Miller took water in it. So that represented the primitive ocean. So the water in the lower bulb represented the primitive ocean. Now, he also created an electric spark in the upper large bulb. So, this electric spark or electric discharge was produced by connecting the electrodes to a high voltage. So, this spark discharge, so this represented the lightning, which Oparin and Haldane proposed in their theory of chemical evolution of life. Now, the water in this lower flask, which is primitive ocean, was made to boil. So when the water was made to boil, it evaporated like this through this tube here. So that represented the evaporation from primitive sea. Okay. Now when this water got evaporated, it got mixed with the gases of the atmosphere where there was electric discharge, which is similar to lightning taking place, and then this water was made to condense using a condenser here. So cold water is made to circulate through this condenser, which would condense the water vapor coming out from this atmosphere, and it made the water vapor fall back into the primitive sea or primitive ocean, and that represented these water droplets represented the primitive rain. So this is how S.L. Miller mimicked or replicated all the conditions of primitive earth which were described in the theory of chemical evolution according to Oparin and Haldane. Now Miller ran this experiment for a few weeks. Remember, he could not run it for as long as millions and billions of years, what would have taken place as it is on the earth, but he ran the experiment for few weeks, and after few weeks he collected a sample of this primitive water here, which is compared to the primitive ocean, and when he observed, analyzed the water, Miller found simple molecules like the amino acids. So amino acids, so Miller found simple molecules like amino acids. So it is believed that many similar simple molecules like nitrogen bases, lipids would also develop, if the experiment is kept to run for many years. Okay, and these biomolecules, so that become a partial evidence. The evidence given by S.L. Miller in this experiment is not a complete evidence for it is only a partial evidence. Because he could not produce a cell from the primitive ocean and show it to the people, but he was successful in partially demonstrating the chemical theory of origin of life by showing presence of simple molecules like amino acids. Now what is the evidence? What are the other evidences for this theory of chemical origin of life? So other people also performed the same experiment like Miller. They found certain other biomolecules like sugar, nucleotides, pigments, fats, etc. So all these support the fact that these simple biomolecules would have polymerized to give macromolecules, and then these macromolecules are, according to the theory of chemical origin of life, these macromolecules would have come together and aggregated to form the first primitive cell. Now, one such structure which is mentioned by Oparin and Haldane in their theory of chemical origin of life was Coeservates. So they said that these simple molecules and other complex molecules can come together due to intermolecular attraction and form a cluster or aggregate called coeservate. So Oparin gave the structures the name coeservates, which are clusters of, so coeservate particles are nothing but aggregates of organic molecules. So another scientist called Sidney Fox. So Sidney Fox, he also proposed a that similar structures would have formed in the primitive ocean, and but he gave them the name microspheres. But nonetheless, coeservates and microspheres are both aggregates of these organic molecules, which is believed to have given rise to the primitive cell. Now there are certain differences, between coeservates and microsphere. So those differences I have listed here. So the concept of coeservate was given by Oparin, whereas the concept of microsphere was proposed by Sidney Fox. Now, according to Oparin, coeservates are made up of complex aggregates of different biomolecules, whereas according to Sidney Fox, microspheres are simple. So he called them as proteinoid microsphere. So proteinoid is a structure which forms when you heat amino acids to a very high temperature. So because they are just made up of proteinoids, they are simple in nature. Coeservates are believed to be less stable, whereas microspheres proposed by Sidney Fox are more stable when compared to the coeservates. Coeservates, according to Oparin, did not divide. So he did not propose them that they were able to divide. Whereas microspheres, according to Sidney Fox, he was able to demonstrate that they are able to reproduce by mechanism like budding. Coeservates, there is no boundary around coeservates in the concept of Oparin and Haldane about coeservates. They did not have any boundary, so they are just enclosed by a water jacket. Whereas according to the concept of microspheres by Sidney Fox, he said that microspheres really have a lipid membrane. So that is what made them more similar to cells when compared to coeservates. So microspheres have more resemblance to cells because of the boundary and other structures when compared to the coeservates. So these are differences between coeservates and microspheres. Now what did these microspheres or coeservates gave rise to? So these aggregates of organic molecules are believed to have given rise to this very primitive cell, which is what we call as protobiont. So the primitive cell, first primitive cell is called protobiont, which is believed to have formed from aggregates of biomolecules like coeservates and microspheres. So this protobiont is later believed to have given rise to the another type of cell called progenote. So protobiont, in many books it is also mentioned as eobiont. So both are same, protobiont, eobiont. So from coeservates and microspheres, you got structure like protobiont, which is first primitive cell. From first primitive cell, we got the common ancestor for all three types of present day cells, which is known as progenote. Now progenote is later believed to have evolved and given rise to the three types of present day cells. So archaebacteria, eubacteria and eukaryota. So archaebacteria and eubacteria, together they are prokaryotes. Okay, so archaebacteria are the very primitive cells. So they are known for their thermostable enzymes. For examples, the bacteria which is found in the hot yellow springs uh, of the Yellow Spring National Park in America is a archaebacteria. So why these bacteria are able to survive in hot water where no organism likes to grow is because when these archaebacteria develop, the earth was very violent. So they know how to survive in hostile or violent conditions or unhospitable conditions. So one feature which enables them to survive in uh, unhospitable conditions is the presence of thermostable enzyme. So one example for thermostable enzyme is the Taq polymerase. So Thermophilus aquaticus is an example for archaebacteria. So like all the archaebacteria, we find thermostable enzymes in thermophila in the bacteria Thermophilus aquaticus. So one particular enzyme which is thermostable from Thermophilus aquaticus is Taq polymerase, which is used in the PCR reaction. Now these archaebacteria are believed to have given rise to the present day eubacteria. So eubacteria are all present day bacteria like E. coli. So the order of evolution of these different types of cells is progenote is believed to have given rise to archaebacteria. So because they were the first uh, primitive cells, they had to withstand the violent unhospitable conditions of the earth. So they were provided with thermostable enzymes. So that archaebacteria is next believed to have given rise to eubacteria, which are present day bacteria like E. coli. And from these eubacteria, it is believed that the eukaryotic cell has evolved. So from aggregates of or biomolecules or organic molecules like Coeservates, Protobiont originated. From Protobiont, progenote originated. From progenote, archaebacteria originated. From archaebacteria, eubacteria evolved. From eubacteria, eukaryotic cell evolved. So this is the order of evolution of these different structures. So if you want here, protobiont is the first primitive cell. Progenote is common ancestor of archaebacteria, eubacteria, and eukaryotic cell. So that common ancestor is progenote. Now, how did prokaryotic cell gave rise to eukaryotic cell? So there are two theories which try to explain us the origin of eukaryotes from prokaryotes. So one such theory is this is endosymbiotic theory. So according to this endosymbiotic theory, so this was proposed by Lynn Sagan. So Lynn Sagan proposed the endosymbiotic theory. Now, what exactly is endosymbiotic theory? So, endo refers to endocytosis. Symbiotic refers to the symbiotic relationship between the two organisms. So, it is believed that cell organelles like mitochondria, chloroplast, so these were independent prokaryotic cells once upon a time. So these independent prokaryotic cell-like organisms, they are believed to have got engulfed into the eukaryotic cell, and they have become a part of the present day eukaryotic cell, and they have a symbiotic relationship between the cell which has engulfed them and between themselves. Now, what are the evidences for endosymbiotic theory? So, if you can see here, the mitochondria is believed to have been an independent aerobic prokaryotic cell once upon a time. So that is getting engulfed here into this ancestral prokaryotic cell. The chloroplast like cell organelles which are involved in photosynthesis, so they are believed to have been independent photosynthetic prokaryotic cell or cyanobacteria before the evolution of eukaryotic cell. So such independent cells are believed to have got engulfed in this ancestral prokaryotic cell, and they have established a symbiotic relationship with the cell, and that is how they have given rise to the present day eukaryotic cells. Now, what are the evidences that support endosymbiotic theory? So, if you see here, the cell organelles like mitochondria and chloroplast, they are bounded by double membrane. Now, what explains the presence of double membrane in these cell organelles? So, let us imagine this is the primitive prokaryotic cell into which cell organelles like mitochondria and chloroplast are getting engulfed. Now, during engulfing, the plasma membrane of the cell which is engulfing these organisms invaginates like this to take them inside. So, this is a very common process which is seen during endocytosis. Now, later on, after completing the phagocytosis, a part of the plasma membrane of the host cell remains around the structure which is endocytosed. So now you have the outer membrane, which is membrane of the host cell, which has engulfed these structures, and also the plasma membrane of the cell which has got engulfed. So those are the two membranes. So the presence of double membrane can be explained like this, which could be possibly because of the process of endocytosis. So the presence of double membrane in organelles like mitochondria and chloroplast are evidence that these cell organelles might have been independent cells which have got engulfed by endocytosis into the primitive eukaryotic cell. The second evidence is susceptibility to antibiotics. So only prokaryotes are susceptible to antibiotics, similar to mitochondria and chloroplast which are found in the eukaryotic cell. So just like prokaryotic cell, mitochondria and chloroplast are also susceptible to antibiotics. So that explains the similarity between mitochondria and chloroplast with that of the prokaryotic cells. If you also see the means of cell division of mitochondria and chloroplast, then they are known to divide by binary fission, which is what is also seen in case of prokaryotic cells. So there is a lot of similarity when it comes to the division also. And also the DNA is circular. So mitochondria and chloroplast, these are semi-autonomous cell organelles. They have their own DNA. And that DNA is a circular DNA, which is also the same found in prokaryotic cells. And also if you see the ribosomes, both prokaryotic cells and inside mitochondria and chloroplast of eukaryotic cells, one can find 70S ribosomes. So the similarity is also in terms of ribosomes as well. So all these evidences, they tell us that mitochondria and chloroplast were independent prokaryotic cells once upon a time, and now they have entered and established symbiotic relationship with the eukaryotic cell, and that is how prokaryotic cells have given rise to eukaryotic cell. Now the second theory which explains the evolution of eukaryotic cell from prokaryotic cell is membrane invagination theory. Hmm. Now, one of the main differences between prokaryotic and eukaryotic cell is the absence of true nucleus. So, what is true nucleus? True nucleus is one which is enclosed by nuclear membrane. Now, how did the incipient nucleus in prokaryotic cell became a true nucleus with nuclear membrane? So, that is believed to have been possible by invagination of the membrane. So, if you see here, this is the ancestral prokaryotic cell. This is its DNA. So it is believed that the membrane, cell membrane of the ancestral prokaryotic cell got invaginated. So invagination is inward folding. So the meaning of invagination is inward folding. So because of inward folding of the plasma membrane, it got circled around the DNA, and that is how a nuclear envelope got created around the naked DNA. So this is how it is believed that by membrane invagination, the incipient nucleus of the prokaryotic cell converted into the eunucleus or true nucleus, giving rise to the ancestral eukaryotic cell. Now, along with these other cell organelles like endoplasmic reticulum, vesicles, Golgi complex, so they also are believed to have evolved by similar invagination of the plasma membrane of the ancestral prokaryotic cell. So these are the two theories. One is the endosymbiotic theory proposed by Linsegger, and the second theory is membrane invagination theory. So these two theories explain how the primitive ancestral cell gave rise to the present day eukaryotic cell. Now this present day, the eukaryotic cell which formed like this gave rise to the first primitive organisms like amoeba, various protistas, etc., which later gave rise to multicellular organisms, and these multicellular organisms by means of organic evolution produced all these present day different plants and animals. So that is the story of organic evolution. Now coming to how the nutrition evolved in these different organisms. So evolution of nutrition in organisms. So why this is important is, so we should remember that the primitive Earth's atmosphere was reducing atmosphere. There was no oxygen in this primitive atmosphere. Then how did these first evolved organisms survived on primitive Earth when they did not have any oxygen available to them? So this happened like this. So initially, to begin with, the nutrition in the first formed organisms, they obtained their nutrition by means of, they were heterotrophs. So they were anaerobic heterotrophs. And they obtained their nutrition by chemosynthesis. So they are anaerobic chemosynthetic heterotrophs. Now these anaerobic chemosynthetic heterotrophs, they later gave rise to chemosynthetic autotrophs. Now later the chemosynthetic autotropes gave rise to anoxygenic photoautotropes. Now all these organisms are prokaryotes. All these organisms that I have written so far are prokaryotes. So initially the first formed prokaryote was anaerobic chemoheterotrope, then it evolved into chemosynthetic autotrope, and later it produced anoxygenic photoautotropes. So these anoxygenic photoautotropes, they contained bacterial chlorophyll. So bacterial chlorophyll, unlike the chlorophyll found in higher plants, does not produce oxygen as a byproduct of photosynthesis. That is why they are anoxygenic. They don't produce any oxygen. So they are with bacterial chlorophyll. Now, next the mode of nutrition evolved into photoautotropes. So these photoautotropes were eukaryotes. So they contained the regular chlorophyll, which could produce oxygen. So we can also say they are oxygenic photoautotropes. Photoautotropes. So these are nothing but higher plants. So this is the order of evolution of mode of nutrition in the organisms from prokaryotes to eukaryotes. Now, when did how old is the Earth? What is the age of the Earth? So, age of universe 20 billion years old. So 20 billion years is the age of the universe, and that is that started with a loud explosion called Big Bang. So that is the age of the universe. Now, what are the important events in the timeline of history of Earth? So suppose Earth had its Facebook profile, then the major events in the life history of the Earth, that is on its timeline, they would appear something like this. So Earth, so this was born 4,500 million years ago. So please remember, 1,000 million is equal to 1 billion. So 1,000 million is equal to 1 billion. Therefore, 4,500 million can also be written as 4.5 billion years ago. So both are same. Okay, so age of universe is 20 billion years. So I have converted the age of Earth into billions here from millions, so which will be 4.5 billion years. Now, when did the first non-cellular life form appeared on Earth. So when I say non-cellular life form, I mean the presence, the formation of biomolecules which should have come together and given rise to the first primitive cell. So those biomolecules are believed to have formed around 3000 million years ago. So 3000 million years is nothing but 3 billion years ago. Now the first cellular form of life, so that appeared 2000 million years ago. So 2000 million years is nothing but 2 billion years ago. And then when did the first multicellular life form appeared on Earth? So the first multicellular life form appeared on Earth 500 million years ago. And then terrestrial plants developed around 140 million years ago. So these are all list of major events in the history of Earth on its Facebook timeline. So next, mass extinction. So we all know that there were dinosaurs walking on Earth once upon a time. So those dinosaurs are no longer found now because they have become extinct, and that extinction of dinosaurs also happened to be the extinction of many different types of plants and animals. So that is a mass extinction that occurred about 60 million years ago. Mass extinction. Now humans have appeared very recently, just 15 million years ago. So that is, those are the list of important events on the timeline of Earth.